Hi, my name is Pedro Barbosa and I'm a clinical research fellow at UCL Queen Square Institute of Neurology in Professor Tom Warner Group. I'll present our findings of a post-mortem study of impulsive-compulsive behaviors in Parkinson's disease. Impulsive-compulsive behaviors in Parkinson's disease includes not only the impulse control disorders such as compulsive sexual behavior, pathological gambling, compulsive shopping, and binge eating, but also the behavioral abnormalities that fall on the compulsive spectrum, such as dopamine dysregulation syndrome and PANDI. Before we talk about the results of the study, I will show you a simplified model of the dopaminergic reward pathway. Under normal circumstances, there is tonic release of dopamine from the ventral tegmental area in the midbrain into the ventral striatum. When the individual encounters a rewarding stimuli, what happens is a fast and large increase in this release of dopamine into the ventral striatum, and that's the way the brain encodes a rewarding stimulus. On the contrary, when the stimulus turns out to be aversive, there is a sharp decrease in the tonic release of dopamine. Parkinson's disease pathology affects the dorsal striatum before it affects the ventral striatum, and therefore it has been postulated that impulsive-compulsive behaviors are a consequence of overstimulation of a relatively preserved ventral striatum. In fact, previous research has shown that excessive dopaminergic release do occur in the ventral striatum of patients with impulsive-compulsive behaviors. However, excessive stimulation of dopamine D3 receptors, which are abundant in the ventral striatum, might also play a part, as suggested by clinical studies showing that the use of dopamine agonists is a main risk factor for the development of impulse control disorders. To better understand the pathophysiology of impulsive-compulsive behaviors in individuals with Parkinson's disease, we have analyzed brain tissue of 31 individuals with Parkinson's disease who developed impulsive-compulsive behaviors in life. The results were compared to 29 individuals with PD who did not have any behavioral problems in life. Firstly, we have used immunohistochemistry to quantify alpha-synuclein pathology and tyrosine hydroxylase expression in the nucleus accumbens, the dorsal putamen, and the dorsal caudate. What we have found is that individuals with Parkinson's disease and impulsive-compulsive behaviors had lower alpha-synuclein pathology in the nucleus accumbens only. There were no differences in the analysis of tyrosine hydroxylase, and these results were confirmed by Western blotting analysis. Secondly, we have used Western blotting to quantify protein levels of dopamine D2 and D3 receptors in the nucleus accumbens. We have used two additional structures, the frontal inferior cortex and the dorsal putamen. Here we found reduced protein levels of dopamine D3 receptor in the nucleus accumbens only. There were no differences in protein levels of dopamine D2 receptors. In conclusion, we have shown lower alpha-synuclein pathology and lower dopamine D3 receptor protein levels in the nucleus accumbens of individuals with Parkinson's disease and impulsive-compulsive behaviors. These behavioral abnormalities could be the consequence of overstimulation of a relatively preserved ventral striatum. Dopamine D3 receptors could be downregulated as a consequence of overstimulation. Increased susceptibility to alpha-synuclein pathology in the ventral striatum of individuals with Parkinson's disease could be protective against impulsive-compulsive behaviors. Thank you for watching.